In this video, we are going to conclude the discussion of translation. How are the messages carried by messenger RNA translated into proteins? How is the sequence of amino acids in a polypeptide being determined? Well, we need to first remember that we have some videos that are introduction of translation, so please do me the favor and watch those. Protein synthesis will occur in the ribosome, and we need to remember that the ribosome is located in the cytoplasm of the cell. Messenger RNA is going to bind the ribosome, then amino acids that are being carried by transfer RNAs will be delivered one by one by transfer RNA to be put together to make peptide bonds. We need to discuss what is that we call the genetic code. And the genetic code is nothing but the sequence of 3 nitrogen bases. So we have first nitrogen base, second pick from here, and then the third nitrogen base that are going to codify for one amino acid at a time. For example, when I say here AUG and I go to the genetic code table, we can find that AUG is either the first amino acid to be put together is the start codon, but it's also methionine in any position on a protein. And if we look for what is CCA, CA, that is going to codify for proline. GCA, we say here GCA, and that is going to codify for alanine. Alanine. GCA, I say valine, but it's not. GCA is for alanine. We change that, so this is alanine. So we have, let's say this one, which is UAC, that is for tyrosine. So we have 3 different nucleic acids codifying for one amino acid at a time. So we identify what is that called a start codon. So that is a start codon. And let's say here, start codon is for AUG. And also we identify 3 different stop codons. So the combination of UAA and UAG, those are stop codon. And I like to remind my students easy ways, so we can remember what is one start codon and what is a stop codon. But what are some rules that we need to follow to understand the genetic code? The first one is that 3 nucleotides, and we have mentioned that, will codify for one amino acid. So that is 3 nitrogen bases for only one amino acid. And that is going to be in the sequence of mRNA. So mRNA is going to be read. Let's say that this is a cartoon for a tRNA that carries an amino acid. So mRNA will be read. So if I have here AUG, that will be making base pair with AUUAGC. So that will be the anticodon that is going to bring one specific amino acid. We already mentioned that this one is for methionine. So that is the first rule that we follow. The second rule that it needs to be followed is that there is no overlapping, that all of these sequences will be read one time. Like when you are going in the train is passing through the tracks one time. So this one, a tRNA is going to read this one. A second tRNA cannot, let me repeat, cannot come and pick CDE. That is going to be not happening because we have what is called translocation of the ribosome. So the ribosome is going to move. Multiple tRNA can combine and read the same messenger RNA, because you have one messenger RNA that is going to be combining the small subunit of the ribosome and a tRNA, and then we will have the second large subunit coming back, and that will continue growing a polypeptide. But once this is read, it's going to be vacant, so another ribosome can also bind and start. But the sequence is going to be read 3 at a time, and it's not going to be overlapping. For example, if this one is codifying for amino acid number 4, another tRNA cannot come and pick these two, let's say HI and J, as a codon, because there is no overlapping in the genetic code. So that is important. Then the next one is when it says that there is no punctuation, and that means that if we have A, B, C, D, E, F, G, one codon is going to read ABC, another codon is going to read D, E, F. It's not going to stop and skip any. There is no skipping of any of the nitrogen bases, and that is what is called there is no punctuation. That will be actually like a dot in the language. So there is not punctuation. The next one, and that is something that the students always remember, is that you have more than one codon codifying for the same amino acid. And I'm bringing this one back from my PowerPoint so you guys can see that combining CU will give me leucine, CUC also for leucine. These are synonyms because more than one combination of nitrogen bases, more than one codon will bring the same amino acid. But remember that the tRNA is very specific for the amino acid, but we have the combination of that amino acid making the ester bond in the 3', but the code is the generic. That's what is the meaning of this word, that we will have more than one codon for the same amino acid. Now these codons are directed. They are different, but they bring the same amino acid with different tRNA. There are at least 20 different tRNA. We mentioned that before, and it's important to remember that we will have what is the start codon. It's going to be AUG, and we call that to be for either methionine or F or 4-mil, that's an aldehyde group on the nitrogen, methionine for prokaryote, and then this one is for eukaryote. And this is to remind you that we have the coding strand of the DNA, the template, that is what is going to tell me what will be the sequence of the messenger RNA. We are going to say again that the sequence of the coding strand is nearly identical to the sequence of the messenger RNA. The only difference is that wherever we have T's, because we have thymines in here, we are replacing those by U's. You have another T's over here, they are replaced by U, but other than that, another T is replaced by a U, more T's replaced by U's, but other than that, we will have that this one will be single strand, and this one is a double strand, 2 strands. The template only is the one that is going to transcribe, and then it's going to codify for only one protein. So we will not have 2 different proteins. It's only one protein. This is just one gene for one protein. And after that, I want to show you a little bit of the steps. But the first thing is for the steps of translation, we will say that the initiation process is going to begin when the small subunit of the ribosome is going to combine with the messenger RNA, and it's going to bring one charged tRNA. If it's a charged tRNA, it means that it has already one amino acid attached. They are going to bind, and then after that, the larger subunit is going to also interlock with the small subunit, creating now we have the site for 2 tRNA to be close together, close proximity, so that we can create peptide bonds between the two amino acids that are attached on top. So what we're saying first is bring one tRNA. First amino acid to be brought is methionine with AUG, is the start codon, but not all proteins will start with methionine. We remember that after post-translational modification, some trimming of the protein, some we will bring some carbohydrates in some cases if it is required, some parts creation of crevices and pockets if it's an enzyme. So post-translational modification means that not necessarily the protein that is going to be folded will contain all of the amino acids that are during translation. Then after that, the second step is for the synthesis of the protein. We have to describe now with this cartoon. So we have the first amino acid methionine that was in the initiation process. So we have two sites. The A site is what is called the acceptor site. This is the one that is accepting a brand new tRNA that is coming charged with one amino acid. Now the P site is called the peptide growing site, because this is where the polypeptide is going to grow. So we have the first amino acid that is methionine. Second tRNA is coming charged, so we're going to create a brand new peptide bond, and then this tRNA is going to leave empty so that it can go back to the pool of tRNAs. I can grab another methionine because this one is very specific for methionine. So that will be for the second step. And then after we have these two tRNAs, they're making one peptide bond, then we're going to have one tRNA that is empty, that is going to leave. So this tRNA is going to leave empty. The methionine is already making a peptide bond. And now the second amino acid that came, that was a glycine, is the one attached to the tRNA. So nothing has changed. But now this tRNA can leave empty to be recharged, to bind to another ribosome. Now the last step, or not the last, but we have the empty that was with methionine is leaving. We have already created a peptide bond between those two. Now we have another one charge coming with an isoleucine. So we need to have first the base pairing. You guys can see that GC, GC, and now AU, UA, CG. So we're going to create another peptide bond. And this is going to repeat many times until the polypeptide is made. How do we know when the synthesis is over? The synthesis is over when some proteins, so we say some proteins that are called release factors, are reaching. And in this point is when the two subunits, the small subunit and the larger subunits of the ribosome, are going to be separated. So we say, yes, you're going to make many copies of the proteins. And remember, this is the same messenger RNA that the sequence is being read by multiple ribosomes. So we have growing many copies of the same protein, so that protein synthesis can be a lot faster than we can see. And then we say here I have some examples of methionine, so you guys can look at the table in your textbook, and I'm going to play a game in a little bit to show you how we can make a protein as a game. And then after the protein is made, remember we're going to have the primary structure. This is what is being made by the translation. And then we're going to have to fold the protein, making alpha helices, making beta pleated sheet, then get into the tertiary structure, and that is using all of the either hydrophobic interactions between hydrophobic amino acids, hydrogen bonds between the polar amino acids, so links between those that are opposite, carrying opposite charges, like lysine and glutamate. That's one example. And then we will look at what is the quaternary structure when we have multiple subunits.